AgTech 360 discusses breakthrough technologies that are impacting growers, businesses, and consumers. Hear from industry and academic experts about what's on the horizon.
So thanks for tuning in to AgTech 360. I'm your host, Adrian Percy, the Executive Director of the NC Plant Sciences Initiative here at NC State University. Now, in honor of Women's History Month and also National Women in Ag Day, it is my pleasure to have Dr. Jean Ristaino join us. We will discuss her journey, her groundbreaking research, and some of the impact of the innovations that she has developed and worked with, like genomic sequencing. Now, her work is shaping the future of plant health, food security, and sustainable agriculture. And we're really lucky to witness Jean making history right here at the NCPSI. So, Jean, thank you so much for joining us.
Well, thank you. It's a pleasure to be here.
Yeah, so let's just start. If you could share a little bit about your journey into the field of agriculture and plant pathology, and what sparked your interest in this field? How did you get involved, and how did you end up at NC State and then eventually at the PSI?
Well, originally I wanted to be a park ranger, take people on hiking trips around the national parks. But applied for a couple of summer jobs and wasn't able to get a job with the Park Service, so I decided to try USDA. Went to USDA in Beltsville. I was an undergraduate at the University of Maryland, and I was hired. And that research experience, that undergraduate research experience, really sparked my interest in agriculture research and plant pathology in general. I worked with a plant breeder who was breeding disease-resistant alfalfa. So we were doing field work, and I was also working with fungi, microorganisms. And I think that undergraduate internship really got me into science. And then from there, I finished an undergraduate and a master's degree at the University of Maryland, went to UC Davis. And interestingly, a faculty from NC State visited and gave a seminar at UC Davis and recruited me. And that seminar interaction with Mike Benson, helped me get the job here at NC State. He recruited me.
Fantastic. So, considering that March is Women's History Month, and with the National Women in Ag Day also coming up on the 21st, I have to ask you the question about, your journey through your career as a woman in a somewhat traditionally male-dominated field, although that ebbs and flows. Have you encountered any specific challenges along the way? How did you overcome them? And what advice might you give to other women looking to break into agriculture or into STEM?
Well, when I started doing undergraduate research at USDA Beltsville, there weren't many women working in ag. There were a few plant pathologists, one or two, but there was a sea of men working in the area. And I looked at that as an opportunity because I felt like there was space for me to do research in that area. And I also had really good mentors while I was working there, and also at UC Davis. When I came to NC State, there were 3 women on the faculty in the Department of Plant Pathology, myself and Margot Daub and Hedwig Triantafilou, who was one of the first women hired in the university as a woman scientist in agriculture. And so I immediately began, as we had faculty interviews and recruiting, I was very active in the recruiting process. And with my colleagues, the best way to change the numbers are to recruit more women into the field, and we did that, over time in our department. Now I'd say we have fewer faculty altogether, but probably 40% are women. So we've come a long way since the early days, but that was important to get more women into the field. Also, in my own lab, recruiting women scientists, postdocs, visiting scientists, just mentoring over the years has been really important.
And, you've had a number of achievements, accomplishments, many of those, and been recognized as well in many different forms. One of the things I'm interested to get your perspective on is recognition as a Jefferson Science Fellow by the National Academy of Sciences, and then more recently with a Fulbright Scholarship, both of which are incredible achievements. And how has these types of awards influenced your work, and have they opened doors to collaboration and different areas that perhaps you've enabled your research with?
When I applied for the Jefferson Science Fellow, the thing that attracted me that it was science policy work. Unlike research, which I've done mostly in the past, I was hired by USAID as part of the State Department and the Bureau for Food Security. And during that year I spent in Washington, kind of on a leave of absence from the university, it was a great experience. From there, over the years, I've maintained my collaborations with folks at USAID, done some consulting work with them, but I also they funded, when I got back here at NC State, they funded a project through UC Davis, a horticulture CRISP project, an innovation lab project, and I was able to roll out phytophthora diagnostic workshops in Central America, and they kind of took a life of their own. After that, I was running them through invitations from different countries, Taiwan, Uganda, Peru. Most recently on my Fulbright program, I rolled out a workshop in Italy. So, one collaboration kind of led to another, and I've continued to collaborate with the CG centers over the year, including SIP, the International Potato Center.
The interconnectedness and the network of global ag research is truly impressive, and you've just basically covered most of the globe in what you've done. And the connections, obviously, that brings to a university like NC State are incredibly important.
The North Carolina Plant Sciences Initiative impacts lives through innovative applications and discoveries. By leveraging cutting-edge research and technology, we address global challenges related to agriculture, sustainability, and human health.
What are some of the projects that you've got ongoing in your lab, and what crops? You've mentioned many of the international crops. What crops are you focused on right now?
Yeah, when I was hired as a vegetable plant pathologist, and I started work early on phytophthora diseases on pepper, but I soon found out that late blight on tomato, it was really problematic in the western part of the state, and also other tomato diseases and potato late blight. So a lot of my research over the years has focused on those crops, but also through some of my international work, we had funding from Dole and worked on bananas, worked on black sigatoka, which is a pathogen that requires 70 fungicide applications to control the disease. They're spraying it more than once a week. It's continual inoculum present. So a lot of these diseases, like late blight, also require a lot of fungicide applications. So some of our work has involved disease surveillance, understanding when the pathogen is present and whether the environment is conducive for disease. That can help growers target when to spray chemicals. We've also developed diagnostic assays for phytophthora diseases. We've started doing whole pathogen genomics to track strains. In the old days, you could use just bits of DNA to amplify and say it's this type or that type. Now we use whole genomes to track how these things spread. And a lot of plant diseases spread with trade. Potatoes move in agriculture, ornamental crops move, and then storms and climate change have influenced the impact and severity of diseases. So, I work on real-time analytics, diagnostics, and pathogen genomics.
So one of the things I know you've focused on recently is looking back and the origins of the Irish potato famine and the pathogen involved in that. So can you tell us a little bit more about that and how you've used some of these new technology advances to understand that better?
When I first started out working on late blight, we were sequencing 400, 500 base pairs of DNA, small bits of the genome, the pathogen genome. Over time, as sequencing technology tools expanded, we were able to sequence the whole genome. The first genome project was a $2 million project funded by a big USDA grant and a team of researchers around the world, and it was a huge project to get one genome. Now you can spend $2,000 or less and get a whole pathogen genome. So what we've done is we've used historical collections from herbaria. Herbaria are kind of like disease libraries of dead, dried plant leaves. You can actually amplify DNA from those specimens, and we've been able to sequence genomes from 140 years ago, not just individual genes, but entire genomes. And using that genomic data, we've been able to track the spread of this pathogen. It moved up from the Andean region into the US and then into Europe and Ireland. And that particular disease, besides founding the field of plant pathology, led to the death and immigration of, death of 1 million people and immigration of another 1 million because of a plant disease. And plant diseases still cause immigration of people. I told you about coffee rust recently. A lot of the Central American folks work on coffee. So migrations of people, when crops are destroyed, people move. But the origins of late blight, the sequences, genomic data has allowed us to really track and define where this pathogen originated. There was controversy between whether it originated in Mexico or the Andes, but all our genomic and ancestry analysis using a lot of computational tools that weren't available 10, 15 years ago have allowed us now to track origins back to the Andes. So that's really exciting. We couldn't do the work without collaborators. I work a lot with Ignacio Carbone, Center for Integrated Fungal Research, and also collaborators from Europe, from Scotland, and from the Herbaria, Kew, Glasnev, and Harvard Farlow's Herbaria. So these collaborations with people and resources have really allowed us to do this work.
Looking ahead then, what are the other challenges that you see in the area of plant health, and what are the opportunities, for different crops, for instance, to improve plant health management?
Yeah, well, we recently had a grant funded by the National Science Foundation called Predictive Intelligence for Pandemic Preparedness. And, in order to we tend to work in the space after an epidemic has occurred, putting the fire out. And what we've done in recent years with our team of researchers here, some in the plant science initiative, such as Chishan Wei, we've developed technologies that allow us to detect early on, before a symptom occurs. And then that data can move into a surveillance system, so you get real-time analytic data. We've collaborated with the Center for Geospatial Analytics to develop a mapping platform called Plant Aid, and that project has allowed us it's scalable to other diseases. So not just Phytophthora infestans, but we're looking at cucurbit diseases and Phytophthora ramorum on trees. Lots of diseases, if you can figure out where they are early on, then mitigation strategies can happen early so things don't spread and get out of hand. And the surveillance tools that we've developed are very useful for growers because the maps are visible on the web. They can look at the internet and look at the website and say, oh, the disease is here now, I need to spray. Or maybe I need to plant this variety because it's resistant. The tools we've developed have helped us track outbreaks, but they've also helped growers, manage their problems more in the field.
I guess the opportunity to anticipate and prevent a disease arriving in your field and maybe use different types of management practices, especially in an era where we see a lot of fungicide resistance, for instance,
Exactly.
is incredibly valuable.
Right now in Europe, there are 2 different fungicides. One that's been used for years, mandipropamid. It's used for lots of different diseases. But there are populations of Phytophthora infestans that have become resistant because of over-reliance on the same chemical. So by understanding, you get the strain and understand its phenotype, whether it's resistant to a fungicide or not, that can help in selecting what to spray and when to spray. In the past, it was always when, but now it's like we can decide when or not to spray at all if the disease isn't present in the area. And so there's big mapping systems in Europe as well. Euroblight is also used, and we have USA Blight and the Plant Aid mapping system here.
And final question, Jean. Coming back to young women who may be considering a career in agriculture or agricultural research, plant pathology, or something of that ilk, what advice would you give them? And, from your experience, what are some of the key takeaways that they could build on?
One thing I would, first and foremost, collaborate with lots of people. You can't do it all on your own. Collaborations with people that know things you don't are very important so you can expand your tool set. And, I've also taken breaks from research and done policy work, and that has really influenced my science because you can understand more how to deploy whatever technology you're developing if you're out there with stakeholders. Our traditional stakeholders would be growers, but also funding agencies, the government, international agencies that might, could benefit from our technology. So, those kinds of collaborations are really important. Networking, meeting people outside the box. Who would, I really working on historic collections, and I've delved into history and things that I wouldn't focus on generally, a plant pathologist wouldn't generally be looking at historical archives. But I think the more you work outside the box, the more interesting your science is, and you can put it into perspective.
Jean, thank you so much.
AgTech 360 shares relevant news and breakthroughs with audiences across the globe. Stay connected and join the conversation by following NCPSI on social media.